Vanakkam. Welcome to this course on Classics in Neuroscience. We have been looking at neural systems, specifically the neural basis of behavior. In the last couple of videos, we looked at the role of hypothalamus and its involvement in feeding behavior. We also looked at neurosecretory cells and how the brain can also be considered as a gland. In this video, we will look at the hypothalamic-neurohypophyseal system, and we will discuss what this means for our understanding of neuroscience. Going by last class's definition of the brain as a gland as well as a computer means that all neurons can be considered as both electrical communicators as well as through glandular communicators, or rather through chemical communicators. Right. Although all neurons are considered both secretory as well as electrical, it was generally accepted that the pituitary gland is the master gland that was controlled by a region, by a nucleus of the brain that lay just above the pituitary gland, called as the hypothalamus. The pituitary body can be visualized as two distinct parts. That is the anterior part, that is the posterior part. So, it was well accepted that the anterior part was glandular, whereas the posterior part essentially appeared to be an extension of cells that originated in the hypothalamus. So, there were nerve tracts, or there were neuronal fibers that extended from the hypothalamus to the posterior pituitary that carried sensory signals from posterior pituitary gland to the hypothalamus. This was visualized as early as the 1890s by Santiago Ramon y Cajal. Also, later Oliver and Schaefer found extracts of the pituitary gland increased vascular tone and blood pressure. So, perhaps it contained some chemical that could increase vascular tone and blood pressure. So, what is the active substance in that extract? That was called as hormone. This led to the introduction and the development of a new field called as endocrinology. In 1928, Kamm identified two main fractions from this extract. First is called as ADH, or antidiuretic hormone. The other, which was somewhat more difficult to extract and isolate, but was eventually isolated, it is called as oxytocin. The location of the pituitary gland in the body is somewhere here, in the head. That location is somewhere here. It required the advanced biochemical methods of the 1950s to synthesize, isolate ADH and oxytocin. It was Lawler and Du Vignard in 1953 who used enzymatic cleavage of glycinamide from vasopressin to produce antidiuretic hormone ADH, and identified that ADH had 8 amino acid peptide structure. Then Popenow and Du Vignard also in 1953 synthesized oxytocin, showing its effect on uterus and mammary glands. The more famous and the more popular synthesis was of course by Sanger and Tupy, who synthesized insulin, although not relevant to the current discussion of the pituitary gland, but methodology, technology was available to do this. So, that was around the time when insulin was also synthesized. Of course, in 1960 there was the synthesis of the nerve growth factor that we saw in the first few chapters, in the first few videos. Bargman in 1951 used the Gomery strain, revealed the pathway of the hypothalamic hormone transportation from the preoptic and paraventricular nuclei to the posterior pituitary using these neurosecretory fibers. From that time, this concept of hormone synthesis in hypothalamic cells and transportation through axons and secretion into the bloodstream has come to be accepted. Still, it was not yet established, it was yet unclear whether the substance that is visualized by the Gomery stains are the same or distinct from the hormones themselves. This still was an active area of discussion. Scharer in the meanwhile showed that there are similar principles of neurosecretion that are found in insects. The first proper evidence for functional properties of posterior pituitary cell came from Eric Kandel. In his study with Alden Spencer, Eric Kandel showed that recordings from the posterior pituitary cells in the fish, he showed that these cells generate action potentials that propagate through the axons to bring about release of their peptide hormones in a manner that is somewhat similar to the release of neurotransmitters. So, the hormones are being released from the neurons as if neurotransmitters are being released from the neurons. But then Eric Kandel was not super excited by this for whatever reason, because he was more interested in the mechanisms of learning and memory, and because this development in the posterior pituitary is somehow not going to help him improve his understanding of learning and memory. So, he somehow did not pursue this line of research and went on to study aplasia in Edward Turc's lab in France. Remember, we discussed this in previous videos while we discussed Turc. So, with this we come to the end of this video. In this video, we looked at the hypothalamic-neurohypophyseal system, and it was finally established that these neurons release their secretory substances or hormones just like other neurons release neurotransmitters. So, there is an action potential that is generated in the neuron that travels through the axon, and then hormones are released just at the synaptic end bulb. Like that, the hormones are released in this case into the bloodstream. With this, we come to the end of this video. Thank you for your attention.